Good morning everyone. We'll continue with the topic cell cycle and cell division with a continuation of the previous class, that is the second period of the cell cycle and cell division. So yesterday I had given you all the definition of the cell cycle, what is generation time and the types of cell division. So already we know that what is cell reproduction. It is a process where the mother cell, the parent cell divides, or there is a division. The cell divides, or the process of formation of new cells from the pre-existing cell is known as cell division. And there are three types of cell division: mitosis, mitosis and meiosis. Already we have learned what is a mitosis in short, what is mitosis and what is meiosis. So in that cell cycle, what is cell cycle? In the cell, series of changes occur where the cell grows, it is metabolically active and prepare itself for the process of cell division. So certain changes occurs in the cells. So those changes is referred to as the cell cycle, or we can say as the sequence of events that occur in a cell with growth, duplication of genome or synthesis of materials so that it can develop into daughter cells is known as cell cycle. And in cell cycle we have studied there are two stages. One is interphase, which is known as I phase, and the other is mitotic phase, which is known as M phase. Why? Again the interphase is divided into three substages: G1, S and G2, followed by mitotic phase. So today class we will deal only with the interphase. Only with the interphase. What all happens in the cell within this period of interphase. So the cell cycle, look at the figure there. So only I will be dealing today with only the interphase part of the diagram. So here the interphase is divided into three sub stages: G1, S and G2. First is what happens in the interphase. What is the condition of the cell when it is in the interphase? So all we can say that between the two cell division, the cell divides. Now two daughter cells are being formed. Now these daughter cells on future or further become the parent cell and divide. So we can define the process of interphase as the period between two mitotic division. We can define the process of interphase as the period or the stage between two mitotic division. It is a period. Interphase is a stage where the cell, or it is a stage or it is a period between two mitotic division. So at the end of the mitosis, two daughter cells are formed. Again, the daughter cells with certain changes is metabolically active and undergo the future or further the process of cell division. So in that phase, so here it is preparing itself for the process of cell division. So that stage where it prepares itself for the cell division is the interphase, or we can say that it is a stage between two mitotic cell division. In this, the cells are metabolically active. The cells are metabolically active when compared to other processes, and especially in the process of cell division. The cells are metabolically active and usually they are are larger in size. So this stage, it is also called as interphase, is also called as preparatory phase or resting phase, where you can see the changes only in the part of the nucleus. You can find certain changes in the nucleus. So this stage is also called as preparatory phase or the resting phase. So if you look at this diagram, look to the diagram over here. This is a chart of the process of mitosis in animals. So what is there in interphase? Already we have learnt the organelles present in it. So in the interphase, you will see that there is a nucleus, nucleolus, the chromatin networks, centrioles, mitochondria, endoplasmic reticulum, Golgi complex, ribosomes, lysosomes and other components which are very active for the further processes in order to go the process of cell division. So you can see that in this phase many products are being synthesized. There is synthesis of many products along with the nucleic acids, RNA, DNA and proteins. In this process commonly there is synthesis of many products. It is mother cell have to divide to form the daughter cells. So it requires more amount of chemical components or the molecules. So it is synthesizing or it is producing. Synthesis means what? Production or produces. It is producing many of the products, especially the nucleic acids. That is the genetic material, the nucleic acids, RNA, ribonucleic acid, DNA, deoxyribose nucleic acids and the proteins which are very important for the cell. And in this interphase you will see that the nucleus is very well organized. It is having a very well organized, and especially in interphase most of the changes occur in the nucleus. It is occurring only in the nucleus with the centrioles with slight changes in the cytoplasm. But main component in the process of interphase is the nucleus. So we begin with the stages of the interphase. In that, the first substage of the interphase is G1 phase. In interphase, when the cell is ready or it is preparing itself for the process of cell division, you will see that it is having a well organized nucleus and other components, and the two main components required in this process are the nucleus and the centrioles. So what all changes or what all happens during the growth phase? First G1 phase. This first G1 phase is also known as, it is known as post synthetic phase. Pre-synthetic phase. So what all happens? We will go through those stages or in the form of points. What all changes occur in G1 phase? So first is there is accumulation of chemical energy that is required for synthesis of DNA, RNA and proteins. It accumulates the chemical energy. So in first growth phase, it is also called as first growth phase or first gap phase or post mitotic phase or pre-synthetic phase, where it is accumulating the chemical components. So here if you see that, you will see that these are the chromosomes, these are the spindle fibers. They all have to be synthesized. In that, the main components that are to be synthesized are DNA, RNA and proteins. So it is accumulating the chemicals that are required for the synthesis of DNA, RNA and proteins, and it is synthesizes all the RNA and the proteins are synthesized in the first growth phase. The RNA and proteins are being synthesized. Apart from this, it is also synthesizes enzymes, nucleotides, amino acids, ATP molecules. To make it clear, some external information can also be given to you all, which may not be in the NCERT textbook, so that the concept is very much clear to you. So apart from RNA and proteins, it also synthesizes enzymes. We know what are enzymes. These are those biomolecules which accelerate the reactions, which are very much required for the process. Many chemical processes occur in the body of a living organisms. Those processes are being accelerated by the enzymes. So in order to carry out the further processes, in the growth phase, the cell also synthesizes these enzymes. And we know that all enzymes are proteins. They are the components of proteins which help in accelerating, that is, they fasten the reactions. For example, if you take the example of salivary amylase or the yeast that is being added for the process of fermentation, which contains the enzyme called as zymase. So these all increases the rate of the reaction or accelerate the processes. Same way, in the G1 phase, the cell produces or synthesizes many of the enzymes and the nucleotides. The nucleotides are the units of nucleic acids. In the lower sections, you have studied about DNA and RNA. DNA contain the nucleic acids in the form of pyrimids and pyrimidines, that is adenine, guanine, thymine and cytosine. So those all the units of the nucleic acids are also being synthesized during this process with amino acids. Amino acids are the components of the proteins. So these amino acids also help in the synthesis of proteins. So and here even the ATP molecule. it requires energy. The cell requires energy to carry out the processes. So in this, it is also synthesizing ATP. ATP is the energy currency of the cell. We have already learned that mitochondria is the powerhouse of the cell. Why it is called as powerhouse of the cell? Because it is involved in the synthesis of ATP, adenosine triphosphate, which is an energy component that is providing energy to the cell to do the life activities or to carry out the metabolic activities. So here the ATP molecules are also synthesized. So it synthesizes enzymes, nucleotides, amino acids and ATP molecules. We can say the generation time taken by G1 phase is around 10 hours with 43% duration of time, or the changes in the cell. So usually it takes around 10 hours to complete the process with 40, and here the cells, the generation time is around 10 hours with 43% of the, compared to S and G2 phase, it is of the longer duration. So in G1 phase, what all happens? Once again we will go through it. There is accumulation of chemical energy which are required for the synthesis of DNA, RNA and proteins. There is synthesis of RNA and proteins. There is synthesis of enzymes, nucleotides, amino acids and ATP molecules, but there is no synthesis of DNA. Still, the DNA is not synthesized in this process. So in G1 phase, these are all the features that occurs in the first growth phase. Second is the synthesis phase. You can look into the diagram that is being given in it. Second is S phase, also known as synthetic phase or synthesis process. Synthetic phase or synthesis process. Here in this, why it is called as synthesis? Mainly the DNA is being synthesized along with the centrioles. So during this phase, there is synthesis of DNA and centrioles. Already we have studied the structures of the centrioles. You can see in this figure, there are pair of cylindrical non-membrane organelles called as centriole just above the nucleus, and during the process of cell division, they produce the irradiating arc-like structures, appearing star-like structures. Those are known as asters. Between the asters, the aster produces the spindle fibers which get attached to the kinetic cord. So for this, you have to go through the nucleus and the chromosomes. You have to be perfect with the structure of the centrioles and the nucleus and the chromosomes so that you can understand the concept clearly. So what happens in the synthesis phase? As the name says, synthesis. Most probably in this process the DNA and the centrioles are being synthesized. Why? There is, or we can say that there is replication there is formation of DNA or the division of DNA, or we can say as replication of DNA with duplication of chromosomes. But remember that there is no change in the number of chromosomes. There is no change in the number of chromosomes. The amount of DNA increases. There is replication of DNA, the amount of DNA increases, but there is no change in the number of chromosomes. For example, if you take in human cell only, already I said that we have around 46 number of chromosomes. Here there is doubling of DNA, but the chromosomes number will be same. The length of the DNA increases, or the amount of DNA increases, but there is no change in the number of chromosomes. It is just 23 pairs or 46 number of chromosomes. Initially if you say that if a cell is having 2C, the number of chromosome is 2C, or there is number of chromosomes. The DNA component is 2C. It is increased to 4C. There is increase in the component of the DNA. Why? Simultaneously there is duplication of centrioles in the cytoplasm. In the cytoplasm, there is duplication of the centrioles. So here you can see that there are usually a pair of centrioles. So that will be doubled, or it is being duplicated. That occurs in the cytoplasm, while there is replication of DNA in the nucleus, with the time period taken is around 8 hours with 33% of changes. Yes, so the time period taken is 8 hours. So once again, what happens in this phase? There is synthesis of DNA and the centrioles. There is replication of DNA and duplication of the chromosomes. There is no change in the number, but the amount increases from 2C to 4C. Duplication of centrioles. Both these processes go simultaneously. DNA changes, or the process of once occurs in the nucleus, simultaneously even the process occurs in the cytoplasm. Both are occurring simultaneously within the cell. So duplication of centrioles is occurring in this cytoplasm. This part of the cell is the cytoplasm. There is duplication of the centrioles in the cytoplasm, while the replication of DNA is in the nucleus, which takes a period of time of 8 hours with 33% of the generation time. It lasts for 33%. So this is about the S phase. Then coming to the next phase, that is G2 phase. Which is also known as second growth phase or what happens in the cell when it is in the G2 phase, or what are the events that occur during G2 phase. So here the synthesis of DNA and RNA continues. Why it is called as S phase? Maximum synthesis of DNA occurs in the S phase. Yes, that will continue even in the second growth phase or the second G2 phase. So there is synthesis of DNA and the RNA will continue. Why? There is formation of macromolecules and those molecules that are required for formation of spindle fibers. So in G2 phase, there are formation of macromolecules. Many chemical substances are being reproduced in our body. In that, some are called as macromolecules, some are called as micromolecules. For example, macromolecules means those which are required in larger quantities. For example, if you take calcium and proteins, we require in larger quantities, while micromolecules which are required in smaller quantities. Take as iodine. Yes, so those molecules are being synthesized by the cell along with the molecules that are required for spindle fibers. You can see in this chart, in the metaphase, you will find the thread-like structures that are being produced by the spindles, which are attached to the chromosomes. So these thread-like fibers are nothing but the spindle fibers. So it is forming those molecules which are required for the formation of the spindle fibers. Here the cell continues to grow. It is continuing to grow, and moreover there is growth, maximum amount of growth with the duplication. It is continuously growing. It also synthesizes, or it is synthesis of energy rich compounds. It also synthesizes some of the energy rich compounds which are required to carry out the further process or to carry out further reactions in the cell. So it is synthesizing energy rich compounds here, and there is synthesis of DNA, formation of macromolecules. It is continuous synthesis of energy rich compounds. As I said that it is helping in to carry out the future process, and the content of the cell increases. No need to explain this, it is already understood. There is content of the cell increases, there is multiplication, of cell organelles. There is multiplication of cell organelles, or we can say that the number of cell organelles cell is having around 4 to 5 mitochondria. It has been doubled. If it is having 4, it is increased to 8. Yes, all the organelles that are present inside the cell, if it is having around 10 or 20 chloroplasts, they are being multiplied to 40. So the number of the cell organelles are increased within the cell. There is multiplication 2 into 4, or we can go in the ratio of 1:3 also. If there are only one component, they are being increased to 3. So there is multiplication of the cell organelles. So the time period taken by this process is 5 hours duration time of 21%, with 21%. So in this class we have studied the 3 stages. Then after this it enters to the process of mitotic division. Just look into the diagram over there. I have explained you the interphase. Yes, up to only S, G1, S and G2 phase. Tomorrow we will continue with the mitotic division, what all happens. So we have studied only the interphase part of the cell division, which includes G1, S and G2 phase. Now after this, the cell is entering into the mitotic phase. Just go through the heading with the chart, that is prophase, metaphase, anaphase, telophase, with the formation of 2 daughter cells. And in the 2 daughter cells, which will grow and again enter the process of interphase, and this cycle continues to continue the life process or continue the cell totipotency of the cell or the generation. So it's a very short class where I have explained you only about the interphase. In the next class we will go what all happens in the M phase or the mitotic phase. So look to the diagram clearly, draw the practice the diagrams over there of the cell cycle, and what are the changes, what are the structures, or what all happens, what are the events that occur in those processes. What are the events in G1, what are the events in synthesis, or what are the events occurring in G2 phase. So in the next class we will continue with the next part of the cell cycle, that is mitotic phase. So all